Okay, this is the third podcast for Standard 2.4. In this podcast, we're going to be talking about the process of cellular respiration and fermentation. In cellular respiration, we're going to be getting energy from our food, and fermentation is just another way of doing that. So when we talk about cellular respiration in general, it's the process of breaking down glucose to store the energy in ATP. So if you remember, ATP is our temporary storage molecule for energy. It's what cells use in order to get their energy. So we have to get the energy out of the glucose, which was made in photosynthesis, and we have to put it into ATP, and then our cells can use that energy source. Cellular respiration occurs in two different places within the cell. It starts in the cytoplasm, and then when there's oxygen available, it will move to the mitochondria. The more energy that a cell needs, the more mitochondria it's going to have. So muscle cells and nerve cells require a tremendous amount of energy to do their work. That is because they require a lot of active transport in order for them to get the impulses that they need to do their jobs. So they have a lot more mitochondria in them than other cells like your skin cells, which don't require a whole lot of energy to do their job because their job is mainly for protection. So when we talk about cellular respiration, this is the general formula for it. And you can see by looking at it, it is very closely the reverse of photosynthesis. So in cellular respiration, we're going to be taking oxygen molecules and combining them with glucose molecules. And from that, we're going to end up with carbon dioxide and water, and those actually are both considered to be waste products. And the main point of cellular respiration is to generate ATP. So in this process, if you remember, photosynthesis was combining carbon dioxide and water to make sugar and oxygen. So now we're going to take that oxygen and the sugar, we break down the sugar, and we're going to get ATP. Now in that process, the carbon dioxide and the water actually end up being waste products of this entire thing. And we end up breathing out that carbon dioxide. But the whole point of doing cellular respiration is to generate the ATP that our cells can use to do their everyday work. Now, the process of getting energy from glucose always starts with glycolysis. So it is the very first step of cellular respiration. No matter what, you have to do this step before you can get any energy out of a glucose molecule. And this step is actually what happens in the cytoplasm. The reason for that is that we are looking at a glucose molecule, and if you remember, a glucose molecule is a fairly big molecule. It's got 6 carbons within it, and we have to actually break that molecule down before it can get into the mitochondria. So in glycolysis, we're going to split the glucose molecule into 2 molecules of pyruvic acid. So we're actually just going to kind of divide it in 2. So where we had a 6-carbon molecule to start, we are then going to break it down into 2 3-carbon molecules. And each of those 3-carbon molecules is called pyruvic acid. So we have our 2 3-carbon molecules. Those molecules are now going to be small enough that they can get into the mitochondria and get broken down even more so we can generate even more energy. Now, during this initial split of our glucose, we are going to gain 2 ATP. So just by doing this one little split of the glucose molecule, we're going to get 2 ATP out of the process. And during this process, we also are going to be using a molecule called NAD+. So this is very similar to the NADH or NADP+ that we talked about in photosynthesis. It's just a carrier for hydrogen, and it's carrying some energy, and it's just used in this whole process of dividing the glucose molecule and getting those 2 ATP. So you can take a look. This is just a little schematic of this. So here is our glucose molecule with your 6 carbons all lined up. It actually takes an input of energy to split it in 2. Okay, and then during this process of splitting it in 2, we end up generating 2 ATP in the end. So our net is 2 ATP, and we also get 2 molecules of NADH. Those 2 are going to move over to the mitochondria along with the pyruvic acid or pyruvate, as they're calling it here. It's the same thing. So all of that stuff that gets made just from this initial splitting of glucose is going to then move over to the mitochondria, where it can get broken down further. Now that is what is going to happen if oxygen is available. Okay, so when there is available oxygen, the pyruvic acid molecules are going to enter the mitochondrion. And from there, we're going to go through 2 additional processing steps of these molecules. The first is called the Krebs cycle, and then the second is called the electron transport chain. But again, this is only if there is oxygen available. So this process is called aerobic respiration, because you have to have oxygen available. Then the pyruvic acid molecules can get into the mitochondria and do what they need to do. The 2 NADH are also going to enter the mitochondria in this case, and they are carrying electrons that are going to be used during the process of breaking down the rest of the glucose. Now, at the end of this process, when there is oxygen available, your cells can net up to 36 ATP from a single glucose molecule. So they can get 36 little energy molecules from one glucose when there is oxygen available. That's really important because there are times when oxygen is not available, and cells have to come up with alternative ways of getting energy, and we're going to find that it's not nearly as efficient. So if no oxygen is available, the 2 pyruvic acid molecules are actually going to stay in the cytoplasm. They don't get to go into the mitochondria, and a different process called fermentation is going to happen. Now in fermentation, we're only going to net the 2 ATP that we make from splitting glucose. So we only get 2 ATP out of this rather than 36 when there is oxygen. This is why living things like humans require oxygen to stay alive, because without it we can't get enough energy to keep our cells going. Okay, so let's take a look, just kind of breaking this down. So here we're starting with our glucose. That glucose is going to get split during glycolysis, and right away we're going to net our 2 ATP. Okay, this is happening in the cytoplasm of the cell. Now when there is no oxygen, we're going to move into fermentation. Okay, and there's 2 different kinds of fermentation that we're going to talk about in a minute, alcohol and lactic acid. Now if there is oxygen available, we're going to move into the mitochondrion, where in the Krebs cycle we'll make 4 ATP, and then during the electron transport chain we'll make 30 ATP. We're also using some NADH during this whole process. So this top part here that happens in the mitochondria is a lot more complex than what happens with fermentation. So we're actually going to start by taking a look at fermentation in this podcast, and then the next one we will go into more detail with what happens in during the Krebs cycle and electron transport chain. Okay, so in fermentation is an anaerobic process. So remember, when you put an A or AN in front of something, it means without. So this is a process that does not require oxygen to occur. So this is what's going to happen when there is no oxygen available. Okay, and basically the whole point of fermentation is it supplies glycolysis with the NAD+ that it needs, so that the process of glycolysis can keep making ATP. So remember, glycolysis is going to split the glucose molecule in half, and every time it does that, it makes 2 ATP. But one of the things that glycolysis needs in order to happen is NAD+. So fermentation is going to supply glycolysis with the NAD+, so it can continually split the glucose and make the 2 ATP. That's its job. This is not, as we said, a very efficient way for cells to get energy, because for every glucose molecule, they're only generating 2 ATP. So this might be fine for a single-celled organism, like a yeast or a bacterium, but for us, who are multicellular organisms and we've got trillions and trillions of cells, getting only 2 ATP for every glucose is just not enough. There are 2 types of fermentations that organisms can use. The first is called alcohol fermentation. And this occurs mainly in unicellular organisms like yeast and bacteria. Yeast is probably the easiest one to think about, and we actually use the fact that yeast does this process to make a lot of our different kinds of food. Because in the process of alcohol fermentation, the yeast end up producing alcohol and also carbon dioxide. Those end up being waste products to this entire process. So we use yeast to make bread rise. We also use yeast to carbonate soda in beer and things like that because they release carbon dioxide as gas bubbles, and the gas bubbles actually is what makes the bread dough rise. Yeast is also used to make alcohol for drinks. Okay, so you also have the alcohol that's found in beer and wine. So for alcohol fermentation, what you're going to have happening here is here's your glucose molecule again. Remember, that's 6 carbons, and it's got to get split into 2 pyruvic acid molecules over here. And to do that, we generate 2 ATP. But in order for glycolysis to happen, we have to have 2 NAD+ available. Okay? So alcohol fermentation takes those 2 pyruvate molecules and changes them into alcohol, which is your ethanol that you get over here. And during that process, we're getting our NAD+ back. And that NAD will then go back into the glycolysis process. So that allows it to keep breaking down glucose molecules. So it's kind of an alternative way of getting energy. We, in our own bodies, actually also have a way of doing fermentation. It's called lactic acid fermentation. And if you've ever had sore muscles, you have experienced lactic acid fermentation. This occurs in some unicellular organisms like bacteria and also in our human muscle cells. In this case, the pyruvic acid that is made from breaking down that glucose is going to be converted into a chemical called lactic acid. Okay, and muscle cells, we use lactic acid fermentation when your oxygen supply is low. So if you've been exercising for a while, you're using a lot of energy to keep your muscles going. And eventually, the oxygen that you're bringing in is actually going to go to your vital organs and not to your muscles. And so your muscles have to start using fermentation in order to get enough energy to keep you going. The more that fermentation they have to do, the more lactic acid is going to build up in your muscles, and that actually makes them feel really sore. And that is why you can get sore muscles after exercising. Okay, so you get this buildup of lactic acid. Lactic acid is also actually used in making some kinds of food products like yogurts and stuff. So bacteria can make lactic acid, and it actually helps to change milk into cheeses and yogurts and stuff like that. Okay, so here's a graphic just showing you how lactic acid fermentation works. It's very similar to the alcohol fermentation. Again, we need to break glucose down through glycolysis, and that's going to generate our 2 ATP. That's the whole point. The 2 pyruvate molecules, the pyruvic acid molecules that are made, are going to get changed over into the lactic acid. And during that process, we're regenerating that NAD, which is going to go back over to glycolysis and allow it to keep happening. So the whole connection between fermentation and glycolysis is that the fermentation is producing the NAD+ that is needed for glycolysis to keep going. In the next podcast, we're going to take a closer look at the actual process of cellular respiration that occurs when there is oxygen available.